flattening out a bit now. Relative to what we were before.
Yeah, well, it looks like we're going to be, according to the map here. Right.
Yeah, it's going to be pretty flat most of the way up, although we could dip out over some of the sides of these ridges if you want as we go up towards waypoint 4.
Sure thing. Go ahead and
Ooh,
push
what's
on
that?
in there again, please.
Nice anemone there.
Is this one of the collections?
Venus flytrap anemones.
This is a flytrap anemone.
Not this one. Okay, good.
Full wide, please.
Full wide.
Sea cucumber.
Okay.
You can see the nodes clearly on that bamboo. That it doesn't
Bridge
have
nav,
any
one more step.
polyps on it.
Yeah, that looks like it's almost just skeleton.
Yeah,
Maybe just skeleton.
it is as far as I can see. Yeah.
Another nice primnoid here.
Is that a different coral under it, or is it growing on it, the yellow?
That's another, it's a yellow coral underneath it. Yeah, there's two corals there together.
Go ahead and push on in there, please.
Good.
Nice shady spot.
Yeah, it looks like that's a little black
Good.
coral
Going a little tighter.
tucked in underneath this primnoid.
All right.
The little black coral is a Staropathes.
A couple of crinoids, it looks like, on that.
And you can see
Very
this
nice.
one's a Narella. We've got our polyps starting to close down.
All right, full wide,
Puck
please.
already. Wide.
What was it?
Silly comment.
Oh, this one's just screaming at
Ooh.
us.
Oh my gosh.
Beautiful.
Rude.
Please take my picture.
Big sponge, even also big brittle stars on it too.
Yeah.
Frantically switching back and forth between my science chat screen and the still camera back here.
Go ahead and push on in there, please. That's neat.
So those orange flecks at the end of its little spikes, are those things it caught? Or do we know what those are?
Go ahead and push on in there tighter.
Like all this little junk?
My guess is those are things that it has caught.
Oh, no, they're moving on it. They're little
Or they could
crisp-
just be
yeah.
little
shrimpies.
shrimpies or...
Go ahead and come partial wide, please.
Push wide. Interesting.
Yeah.
Is that a Walteria sponge?
Yes, that's another Walteria sponge. Steve says those are benthic ctenophores.
Full wide, please.
Chalphelius.
Another one of those large bamboo whips.
Oh, another one that's really probably close to 2 meters long.
Is that a Victor Gorgio? Can we zoom in on that
can we
guy?
zoom that?
Sure.
We had a request for a zoom earlier, and then this is the first one we've seen since the request.
Whoa, this is cool.
Yeah, Victor Gorges are awesome. Let's get us set up, sure.
I want it. Whoa.
All right, go ahead and push on in there a bit, please. Keep it heading around a little bit.
Very nice.
Is that I don't know, alternatipathes?
Go ahead and push
Is that
on in
how
there
you say
again,
it?
please.
Behind
This
it?
is Victor Gorge, or behind it?
Yeah.
Oh, I'm not seeing.
There's like a stock looking thing behind it.
I'll look when we zoom
Nice
back out.
focus.
Sweet.
Thanks.
This is Victor Gorgia,
Is that one big one?
possibly Alba.
Wow.
One big star or a bunch of little ones?
No, that's a big stuff. You can
That's
see
a
it's
big one,
center.
right?
Yeah.
That's so beautiful.
Awesome. Go ahead and come partial wide.
Partial wide.
There was something behind it you wanted to take a look at?
Oh, just to the right there?
Yeah.
Oh, that's the Umbellapathes.
Oh, okay.
All right, full wide, please.
Full wide.
That we sampled about an
Reach
hour
nav,
ago.
one more step.
That one just
Cool.
happens to have 5 leaps instead of 3. Oh,
Cool
look at this.
candelabra.
Wow.
Just when we need to move, buddy.
So cool.
Look at you.
That's a perfect
This
shape.
is a perfect I4 clade
Oh
bamboo.
my gosh. Can we take it?
Get the bubble cam out of view there.
It's like a tiara type deal.
Yeah.
And there's like some sort of sea spider.
Go ahead and push on in there a bit, please. That's good.
Wow.
Oh, nice. And a nice crab hanging there.
Ooh.
The back of this guy is cool. Wow. Go ahead and come partial wide.
Partial wide.
That's nice.
I suppose we can float up just a little so I can get it nice and centered in the still cam.
Yeah, sure.
It's unreal.
Let me know when you feel like it's centered up there. Oh, I will dust cloud behind us, but
Just a little higher.
a little higher.
Yeah. Since that's down on the porch.
Out there.
One more little bump up. It'll be nice and centered.
How about there?
Great. Thank you.
Yeah, sure thing. Thank you for driving the still cam. All right, now boogie. I'm gonna come up a bit there on the delta.
Yes.
Thank you.
Wait, was that an I4 or an I2 bamboo coral?
That's an I4.
Oh, gotcha. Thanks.
Nice primnoid there.
That's weird.
What's up?
It's like a bruise on my leg for some reason.
I bump into stuff all the time.
Yeah.
So where did that come from?
If it makes you feel any better, I fidget around so much during these watches that for the past 3 times I've ended with my shoelaces untied.
Wah.
Kevin, on our last seamount, the Geo, last dive, did we find any carbonate?
I haven't cut the rocks open yet, so I'm going to do
okay.
that after this watch. We'll see. I mean, it was definitely clearly carbonate. Structure-wise, yes. You can see the transition. We went from the steep walls that had these like thick, blocky layers. So it was a clear morphological transition. Whether we have any samples, I haven't checked.
Breach nav, one more step.
Huh, this guy looked like he got split asunder.
Yeah. Someone gave it a bad haircut.
I need to do it. I need to get a haircut.
It's a calyptophora there. Wonder if a boulder came down from above and landed on it.
Maybe.
Oh.
The weight of too many brittle stars. Split it.
Marshall.
We got another good coral coming up. I can see it in the still cam.
The one to the right?
Straight
Kinda-
ahead.
Straight ahead, just upslope from us.
Ah, Reg. Yes, you have good eyes on that still cam there.
What all do we have here?
Put them cushion in there,
I think
please.
this is another calyptophora. Just like the one that we saw, but this one is undamaged.
Ah,
Has no, hasn't been to the barber shop yet.
Probably calyptophora lyra, says Steve.
Very cool.
and what are the yellow ones off to the left?
This is that same plexaurid we were looking at down below. And there's a Stauropathes behind it. So we've got 2 different yellow
At the
species
stop,
there.
it looks like it's taped. It looks like it's just taped to the rock.
Kind of does. You do a quick full zoom there, please.
Full zoom.
Gorgeous.
Nice shot of the polyps. Very nice. Cool. Go ahead and come wide, please.
Full wide.
Do you want another digital stills of this one too?
I got some good ones as we came up to it.
Roger.
Good little chrysogorgia here.
I'm good.
Bridge nav, another step.
Can we zoom in that call while we wait?
Yep, sure thing. There's actually another one here too.
Oh, and a pickle.
Ooh.
You want to do both of them while we have
Yeah,
some
let's
time?
do the pink one because I'm calling the shots, so I'm going for the prettier one.
Roger. We won't tell that to the other coral.
I like my colors brightly colored.
Go ahead and fish on in there, please.
Oh, the other one will have a gold skeleton underneath.
Oh, fancy.
Oh, well, we have to go look at both of them.
I was gonna check that one out now.
Little bubblegum coral here.
No, we have to. It's a nice one. All right, go ahead and come wide.
Color wide.
It's got a little chrysopathes next to it.
It's already 2:30. Wow.
Yeah, let's
Time
watch
flies when there's lots of corals.
this. And sediment. Last night was all right.
Time flies when there's a Gorgias.
Especially when there is a pair of gorgias.
Surely one's enough.
Go ahead and fish on in there again, please.
Nice little Norella.
Bridge nav, another step.
Go ahead and throw in some more zoom there, please. Oh, they're all closed, aren't they?
Yep. Now you can see their nice polyp orientation pointed down. Oh, just got Steve chiming in. Yep, Norella.
Go ahead and come a little wide there.
Go wide.
It's a nice base. A wide, please.
Go wide.
Yeah, these ones, when they don't have the coating of sclerites over their skeleton, the skeleton actually often has a metallic gold sheen to it.
Oh boy. Oh boy.
Chris, how far have we come in this watch?
Say again?
How far have we come on this watch?
You sure you want to know the answer?
I know it's not a lot, so that's what I want to know.
50. No, no.
Let's see. Where did we
62.
start? We were there.
Go ahead and
That
come
was
on
our
up in the
first sample,
delta,
so
please.
430 meters.
Roger. So total, we've come about 1 kilometer since we started
Well,
at a visitor's bay dive.
Is that
Sure.
a mushroom in the cave? Or, I don't know.
I think that was a
No, we went further than a
feather
kilometer.
star in the
They
cave.
Feather star.
they did 800 meters before us,
Roger.
Who's this?
so we've done about, yeah, a little over, I guess. 1.3 kilometers, something like that.
All right, and it looks like between waypoint 3 and 4, it's not.
Oh yeah, we should be able to pick up the pace here.
Yeah.
how many work
It's a
factors
little
is that?
Chrysogorgian.
It looks like for a while, actually, this right will be
Mm.
a little bit flatter.
Okay, sounds good.
Chris, could you talk about a little bit about the 3D map that we see on channel 3? How are we getting the position data from Herc?
Oh. Well, that's a pretty deep question. So we get the navigation data a couple ways. We have a couple sensors. So we have the USBL, which is the closest thing we have to a GPS underwater. It gives us our position to within a couple meters. Unfortunately, that's not good enough to make high precision maps like we see here. So we need to fuse that data with the DVL, which measures our velocity very precisely. And if we know our velocity and our heading from the orientation sensor, we can sort of fill in the gaps and sort of interpolate in between the coarse USBL pings. And with that, we're able to get our nav solution down to maybe 10, 20 centimeters, which is kind of good enough to make these types of maps. Bridge nav, one more step.
And these sensors are on Hercules?
They are, yeah. So if you look at the screen, let me see, I'll pull them up. So
Iclinoid over to the left on top of a...
we have it out on the satellite, Sat 3 or something?
We do, Sat 3. Yep.
Okay.
Channel 3.
Let me turn off the water column view here for a second so we can see a little better.
Whoa,
Whoa.
that's crazy. Sorry.
Make that into a shirt.
Okay, that actually didn't make the water column view go away, so. Yeah, so the sensors are actually on Hercules. So the DVL is right here. You can see it hanging off, and that has 4 beams. It basically measures how fast we're going and along the track of the vehicle and across the track of the vehicle. The USBL is up here, right here on the top, and this shows acoustically is able to recalculate Hercules' position directly. And inside here, which is going to be a little hard to see, right in here, this little can, is the octans, which measures our roll, pitch, yaw, and the accelerations in X, Y, and Z. And if we, with some fancy math, if we fuse all those things together, we're able to get a pretty precise position.
Very cool.
Oh, Chris, you could make a T-shirt out of that.
Out of this?
It's all
Yeah.
out of concert.
Oh yeah. See, there's our track, our mini map.
That's cool.
Yeah, we've been a little when we're going up slope like this, like the Norbit is very close to the bottom at all the time, so we don't really get awesome Norbit data, but we get something. And also the DVL has not been happy on this terrain.
What does DVL stand for?
Doppler velocity log. So it actually measures velocity by measuring the Doppler shift between the forward-facing beam and the aft-facing beam. And
cool.
it can measure the velocity sort of amazingly precisely.
Can we get a zoom on this call?
Yeah, sure.
Thank you.
Go ahead and push on in there, please. Go ahead a little tighter. Nice, nice focus.
Yeah.
Like a little bonsai coral.
Time to grab a snip of this one.
We're
Right,
off
Close-ups.
SBL.
We're looking to grab a snip of this one if possible.
Yeah. Roger, full wide, please.
Full wide.
Go ahead and, I guess we only have 4 meters left in that ship move, hey?
Taken?
We're gonna take a sample here.
All right. Yeah, the ship move is done.
Roger.
I can back up if you want, but
Nah, all good. Snip and slurp, rinse, repeat.
Yep, that sounds perfect.
Roger, roger.
And we don't need the whole thing, just a little branch should be just fine.
Awesome.
Got to put on Herc's garden ending gloves.
We're gonna do a little bit of pruning.
Oh man, makes me miss my peach trees.
My plum trees at home should have fruit on them by the time I get back.
Oh, that's so exciting. I decided I was eating random peaches
Go ahead and push it in one
during
more time.
during the pandemic, and then I decided just to put it in a pot. Now I got 2 peach trees growing.
That's a pretty good one, though. All right, full wide, please.
Full wide.
Can I go ahead and put up the bucket cam, please?
Yeah.
So this is a plixaurid coral.
Go ahead.
And the thing with plixaurids is they often have a lot of overlapping morphology. So we're going to take a little snip of this one to see if we can make sure we confirm that species ID is what we think it is and make sure that it's in our genetic database.
Every time. What's going on here? There we go. All right, Tim, go ahead and push on in there a bit, please.
You also got the
That's-
still cam if you want like a different perspective as well.
I don't think I'm on still cams, but come a little wide, please.
Full wide.
A little wide, just so we, thank you. Too much, way too much. Let's get this a little bit smaller. Stand by. All right, is that about, it's kind of a lot.
That looks like great.
Looks good.
Yep.
Trying not to get the other branch.
Yeah.
As well to take both.
There we go.
All right, so that looks like a small snip. Is that good for you guys?
Yep, that's
Yep.
great.
All right, have you zoom in on this guy a bit, please?
This one's interesting because usually they're kind of like neon yellow almost.
Ow.
This one's kind of golden.
Okay. Full
Yep.
wide, please.
Full wide.
Get my arm all straightened out here. Oops, sorry. right, go ahead and push on in there a bit, please. That looks great. And then go ahead and start your slurp there. Want to get kind of
You're
full jam.
kind of far off, but yeah.
Yeah, go ahead and start it up though. Great. Okay, ready? Can't terribly see it going, so I'm gonna open it slow. All right, I think that went in. At least we got one piece in. All right. Looks like there's one other piece here. So keep it on full jam. And then I'm gonna open again.
And there it is.
Yeah, looks like there's one more in our claw here.
Yeah.
Full wide, please.
Thank you.
Just to confirm, that went into slurp jar 3.
That's correct.
Perfect, thanks.
Go ahead and zero the slurp.
It's zeroed.
Oh, interesting.
Uh-oh.
Where did it go?
I saw the bigger piece go in.
Oh, we tried jostling them back and forth a little bit.
Yeah, I saw it going in too.
Yeah, there was a big piece in there. It might just be stuck up to the side of the filter.
It better be. It's got to be,
Do you want another piece?
just in case.
I saw it go in there, so I'm sure it's in there.
Okay. Yep, sounds good. Just surprised it's not like jostling itself down.
Yeah. Yeah, it's got to be getting stuck on the filter.
Roger.
All right. All right. Ugh.
So you guys are sure you don't want another little snip?
Yeah, I think that one was just fine. I'm confident it went in there. I saw it go in there, and the little piece that's in there would be plenty for us to do genetics with, so.
Okay. Roger that. All right, Chris. I'll get actually a little ahead. Good. I'm gonna go ahead and look at the porch there, please. And then give me the starboard bio box. Please and thank you.
This is flat.
Yeah, we made it up to our third waypoint, which is essentially the top of a little flat knoll on the ridge. Then we'll get back on a ridge going towards waypoint 4. Still getting some corals up here, just not super dense.
Bridge nav, 20 meters, 170.
If you were backpacking in the mountains, this kind of knoll would be the great spot for your campsite with a view.
Yeah. Some very nice sheet flows.
Are we able to tell the current where we are right now? What the current is like?
For that, we usually take our hands off the sticks. And right now my hands are off the sticks. So there's really no current here right now. Nothing really going on. Probably why it's devoid of a lot of corals.
So I think as we go towards waypoint 4, if it looks like this with not a lot of current, we might want to go south to see the lip, kind of on the cliff. A little more south.
A little more south.
Yeah,
Like here?
yeah, like
So like
moving along.
southeast, or you
Yeah,
want to
yeah,
go along this?
southeast.
Okay.
Yeah.
Oh, I mean, just lateral across.
Yeah.
Sounds groovy. Let's do that.
I got to zoom on that coral right under the lasers.
Yeah, sure thing.
It's another one of these bottle brush chrysogorges, I think.
Give me a little bit of scope there, please. I think I'll just actually set it back here. We're at the end of our leash, so I'll just do this instead. Go ahead and push on in there, please.
Ooh, not a chrysogorgia. That's a black coral.
Sorry, stand by. Go ahead and push on in there more.
Little bitty guys. Ooh, bitty little. You can see the little tiny polyps there.
Yeah. Go ahead and come a little bit wide.
A little bit wide.
Don't see any sub-branching off of those pinnules.
If you want to just keep the moves, either, I guess until we get over to the steeper bits, we can just do continuous moves at like point. Let's do 0.2.
Warp factor 2, aye.
Warp factor 2.
I'm gonna do it, watch.
Steve and I are both guessing this is heteropathes.
Breach nav, 150 meters due south, warp factor 2.
Full wide, please.
Full wide.
0.2.
Odio's like, my God. I didn't catch that one.
That was just either here we go.
He's keeping it professional. It's International Seafarer Day today, right?
Yeah. Yeah.
Oh, yeah.
This is when I suppose the song Brandy by the Looking Glass would be appropriate. No, because it's International Seafarer Day.
Yeah. It's an appropriate song.
Another Walteria.
Are we in a position to grab a rock?
Yeah, we are.
It looks like some nice angular ones floating around over here.
All right, let's do it. Let's do
Or
quick
not
grab.
floating around.
Oh yeah, well, sinking. Staying in place like a proper rock.
We're looking for a bigger one.
Yeah.
Raj. How about this one right in front of the slurp? You
Yeah,
want
that
angular?
one's pretty nice.
Raj. Hey, how about it?
So. You talking about this guy?
Yeah.
Yeah.
Let's see, 55. I'll grab and then we'll switch. And I get set up for the starboard biocam there.
Yeah.
Yeah. That was all Kevin. Go ahead and push on in there, please.
Actually, is.
Scaling lasers. All right, keep on going. That is great.
I wish every seamount was this well behaved. This is a fantastic rock.
Awesome.
This one tells a story.
It does. All rocks tell stories.
All right, full wide, please.
Yeah.
Full wide.
All right, gonna look over at the, yeah, you do. Sick. This one's going into E, correct?
Yep, E is empty.
Go ahead and switch over salvos, please. Awesome.
It's a shame you can't like put the salvos out on the channels.
That's true, that'd be cool. All right, you can go ahead and pop it in. Thank you. Danke.
Thank you, thank you.
Anything else you guys want while we're here?
Can we go look at that coral that's in the back corner there?
Sure thing.
This one?
Yep.
Yep.
You want it in this salvo or
Yeah, go ahead and change salvos over.
change it?
Yes, please. Coral back corner. Would you mind illustrating it again, please? Raj. Would you mind putting up the port rail cam there too, please?
Oh, yeah.
Would you mind giving me the porch cam on top? Thank you.
I think this is going to turn out to be just another one of those plexaurids we just sampled. But nice to get a nice look.
Go ahead and push on in there, please. Not really the view we want, is it? You guys want a better shot of that?
We got some good shots as we approached it. So
Roger.
we're good. Thanks.
Full wide, please.
Wide.
You know what that little black spot is on the rock?
Maybe it was a shadow, hey? That one back there?
Yeah, I don't know that. Yeah, is it a shadow or is it?
Go ahead and push on in there quick, please.
Probably just a shadow, but we'll see. Yeah, I'm guessing it's just sharp. Yeah.
Sharp rock. Full wide, please.
Full wide.
All right, I'll get you out ahead here.
Something like a red stalk.
Looks almost like a paragorgid or that had tipped over.
Yeah.
Sorry, did you want to see the red stalk?
Sure.
Oh man, I need to take out a wrap. All right, go ahead and push on in there, please.
Yep. Poor coral. Looks like it broke off its holdfast.
Yep, looks like it broke off. Sometimes they can reattach.
Oh.
All right, full wide, please.
Full wide.
I want to take care of a wrap real quick. So we're going to just spin in a circle briefly.
So how would they reattach? Would they move or the current?
They can basically grow their tissue down in that direction. So sometimes with the paragorgias, we'll see ones where like you have kind of a stump of a base and then another big branch grows out of it, or they'll be kind of sideways and then suddenly be growing vertically. So it depends on the local conditions. They can't all do it, but it is possible for some of them to just basically redo a nice hold fast and keep growing. That was a nice big primnoa down there.
All right, we are back on track. I'm gonna pass this over pretty soon to Michael.
Dude.
Roger.
What is our move? 170?
180.
Oh.
What I'm gonna actually have us do while we get set up is I'm gonna move Argus this way off the cliff, and Herc's gonna stay here so that we can stretch out the other way now. You know what I mean? That way you're not on top of me, especially when we go to the cliff face. So we'll actually hold off here for a minute and then stretch out so that Atalanta looks at around, looks north. And then I'll just trail along that way. As long as the bathy is correct. I'm good. Raj, go ahead. All right. Back row, we're just doing a ship maneuver to get the vehicles in the right orientation as we go down slope and also approach that wall.
Excellent.
So should take another 2 minutes and we good.
Picked a beautiful parking spot.
Yeah, not a bad one.
So the slope, obviously, is going to be pretty gentle from waypoint 3 to 5. So if the life is pretty sparse, we might start increasing speed to try to get upslope to the summit. But for now, let's just see what we find.
Sure thing. Yeah, the orientation that we have right now is in anticipation of popping down slope and looking at the sheer wall.
Okay, excellent.
But if you guys want to go along the flats, then we can change our orientation and kind of go fast. But in this orientation, we go slower.
Okay, yeah, let's start looking at the wall first.
Okay. Sounds great. All right, we're going to do a pilot swap here. Oops, sorry.
While we're doing that, what's one of your favorite facts about any sea creature? So a random fact to any of our scientists.
I'm going to have to think about this one. Favorite random sea creature fact.
I think the nudibranchs that incorporate the photosynthesizing organisms into their, what are the spikes on their backs? That's pretty cool.
So we're in the right orientation now for you to lateral and back down the slope here. And yeah, so you're clear to go ahead and start moseying down. And then the cliff's gonna stay ahead of Herc and then always like furthest away from Atalanta, so we don't hit Atalanta onto the slope. So you're clear to just kind of stay within that frame there and then just kind of back and lateral.
I should say it was a variation on that nudibranchs that incorporate photosynthetic organisms into their cirri. There are a couple species of nudibranchs that eat hydroids and can take the nematocysts from hydroids and incorporate them into their cirri and make themselves toxic.
I don't know if this is a fact, but I love the crabs that put little anemones as a hat to protect themselves. I think it's a fact that that's adorable and clever.
Gotcha.
So I used to work on Alaskan salmon, and I like that there are different salmonids that will decide that they no longer like to go to the ocean. They just want to stay in freshwater lakes for all their lives. And the most well-known of these is the opposite. A steelhead is a trout that decides to go to the ocean. But in sockeye salmon, there's a variation of sockeye salmon that do their entire life cycle in lakes called kokanee.
Mine is more of like a favorite deep sea creature. I don't actually know that much about it, but it's called the telescope fish. And I actually just discovered it yesterday on a Google search. And it's called a telescope fish because they have like tubular, like lens-shaped eyes that help them have like really good vision in the deep sea, and it helps them see bioluminescence from a distance. So it's a cool adaptation, but they also just look absolutely ridiculous, and I love them.
That's great. There was an enemy, it looked like, eating that bamboo coral at the base. that we just passed.
I missed it. Just over the lip and we're back into all of this Metallogorgia.
Just keep moving back.
Oh, I've got a weird one. There's sea spiders don't actually have a respiratory system. They oxygenate their tissues by flushing their guts around. So they like sort of squeeze and flush water around with their the guts that run down the length of their legs.
Huh, that's crazy.
Yeah.
There's an echinoderm eating a bamboo as well. All these bamboos are getting eaten. Oh, thank you.
All right.
Thanks, Jess.
Jess, you rock for this chocolate.
Whatever keeps us awake. In addition to the cool coral sightings. Nice. Mm. Alright, so now it looks like there's some feature coming up behind Atalanta, so I'll have you, we'll do this for another minute or so, but then I'm gonna have to come up a little bit, so I'll have you go to the left-hand side of the screen where the slope starts to be more prominent.
Do we know how long or tall bamboo corals can get?
I have seen a couple of them that are a couple meters long, and I've seen some that are a couple, 1 to 2 meters across and high. And I think they can get even bigger than that. I think Steve has a good photo of one that is kind of towering over Hercules from Puerto Rico.
Looks like the largest bamboo coral was 9 meters, or about 30 feet tall.
That's pretty tall.
Yeah.
Steve has chimed in that his photo is from the Cayman Rise.
Little pink corals over there.
And a little primnoid next to one of them. And the Paragorgia bubblegum corals can actually get huge, huge. There's some really famous photos from New Zealand of ones that are tree trunk size in diameter. I think that's a Baltusina sea pen there.
Can we get a zoom on it?
Sorry guys, if it's all right with you, we're just doing some brief setup training for this.
No problem.
Yeah, thank you.
Ooh, there's a nice coral. So for anyone following along at home, you can go back and watch previous dives from this expedition. After a day or two, they get posted to YouTube. And Kenneth Sulak, who is one of our scientists ashore and a fish specialist, has just chimed in with some of the identifications from fish we've seen over the last couple dives. He says we've seen a nice Chaunacops coloratus, a species inhabiting continental rise depths and probably identical with the Atlantic form. We've also seen a couple cusk eels, a tripod fish that is probably Bathypterus ventralis.
Mexico.
So where is the oxygen minimum zone in this part of the world? What depth?
We're looking.
Sorry. I guess I'm asking because that seems like a place where you wouldn't really find a lot of fish.
No, you don't find really anything in oximo minimum zones. You can get anoxic enough that you can actually precipitate pyrite directly out of the seawater in those kind of areas.
What's pyrite principally
Sorry.
composed of?
Fool's gold, so it's an iron sulfide.
Iron sulfide. Okay.
Yeah, it's not necessarily stable at Earth's surface, but in really anoxic environments it can form. And so when you see it in a sediment column or a sediment core, you know you're in anoxic conditions.
So I just found a paper. It says in the tropical Pacific, oxygen minimum zones are located above intermediate depth waters, defined here as the 500 to 1500 meter water level.
Okay.
So shallower than where we're operating.
Thanks for looking that up.
Also, side note, pyrite sometimes forms in perfect cubes, and it's really cool. You should look it up.
In areas like the Santa Barbara Basin, there's, depending on the glacial cycle and climate, global climate, it will go from axic to anoxic. So you'll see in sediment cores, there'll be little burrows made by critters called bioturbation. And then once you get into an anoxic period, there's no disturbance of the sediment. It's perfectly layered with nothing disturbing it, and get little bits of pyrite, and then it goes back into being all disturbed due to bioturbation. So it shows that nothing on the seafloor is really living. There's a big Walteria sponge on the left.
That's so cool.
Zooming in a little bit.
Got a Parentopathes black coral, and just to the left, there was also a little Chrysogorgid.
Zooming in. A little bit more. Zooming out.
So did you say you know a scientist who's studying, I guess, the relationships between the brittle stars and the corals or the sponges?
Not actively right now. I know a couple folks at Woods Hole have been doing that in the past, but I don't know if that they're actively working on that at the moment.
I have a more broad question for anybody. I think that a lot of people's motivations for going into like ocean science is because it has direct links with studying climate science. And I was just
Sorry,
wondering.
I can barely hear you, Jess. Okay, so we want to start heading back towards, like across the slope, yep. So still south you want to do. What are you thinking?
I can't hear anything Jess is saying.
Yeah.
No, sorry, I was off SPL.
Whoa.
Oh,
We were
okay.
in,
There you are. You're back.
So what is that?
So, Chris, go ahead.
Yeah, so do we want to sort of continue along the slope towards waypoint 4? Do you want to go down a little further, or what do you want to do?
I think we just go straight towards waypoint 4, yeah.
Roger.
Roger.
If we don't see much, I think eventually we'll just turn around and start speeding towards waypoint 5.
Okay.
Rog. Okay.
Bridge nav. 20 meters, 225.
Okay, so now this is a different orientation for you. So the difference is here is that originally if we were going to go skate along this contour, then we'd want to have this lateral action going on, and then you stayed out, kind of similar to what you're going for right now.
Yeah, yeah.
Now that we're just going to go kind of head over to waypoint 4, I'll maintain the heading to waypoint 4, and then you'll just stay out in front that way instead of any lateral action along the contour. That kind of, that jives, yeah?
I think so.
Okay. Yeah, so one is like making sure that we're just like skating and maintaining the sonar, like the right rings across. And in this case, now that we're not skating anymore, we're just going to go straight. We'll just change orientation, and then I'll have you get out in front over here. So you can go ahead and start making your way that way. And then I'll change my heading. And Chris,
Yep.
what's the heading? It's going to be
224.
224, Rog. So you can go ahead and start making your way over here somewhere.
So start going forward?
Yep, all good.
What was your question, Chanel?
Oh yeah, I forgot I was even asking something. Yeah, I was going to ask, so I think a lot of people go into ocean science because it's like really closely related to climate science. 70% of our Earth is oceans. And I was just wondering what kind of ways you all translate what we're doing here in this control van and seeing these samples to like advocacy or ocean science, or trying to raise awareness and help combat climate change.
Short answer, I don't. I support all that, and I try to teach people how cool these features are and how important it is to preserve them, but other than that, I don't have any direct links personally, but I know plenty of people who do.
A lot of the areas we are looking at in the really deep ocean are somewhat stable, so they don't see the big effects that you see in, say, shallow water tropical environments yet. But we expect those climate effects to eventually percolate through down to these deep water habitats. And then some of these corals that I work on off the Pacific coast in Alaska live at much shallower depths, and they're quite temperature sensitive. And so in those habitats, we are starting to look at how they interact with temperature and what the impacts might be to them as the water is closer to the surface warm.
That's cool.
Kevin, you said you know scientists who, maybe geologists, who kind of connect the dots between like climate change and geology, depending on what they're studying?
Yeah, it's very common for, well, it's a major field of geology. It's called paleoclimatology. And you're looking at things like sediment cores, and of course ice cores, but in the deep seas, we're looking at sediment cores. You can see how deep ocean currents change, how sedimentary regimes change, and then you can also record changes in ocean chemistry by looking at the little fossils. So all this marine snow, there's plenty of fossils from little critters living on the surface of the ocean. And when they form their shells, they have distinct stable isotopic compositions, and they record them, and then when they rain down, they preserve that through time. So you can actually see how ocean chemistry changes and link it to see what effects rapid and slow changes in climate have on oceans and the deep oceans as well.
Over here.
Yeah, just away from the sponge though. Yeah, that works. Back row, we're gonna drop a weight.
All right, sounds good. Looking around the saddle, there's so much of this Walteria. You could almost call it the Walteria Wonderland.
Yeah, it seems like we're getting less density, but we have single
Nice.
critters that have really long stalks, and it's like if they find a really good rock, this is a good spot for them.
All right.
How is that backwards, sideways flying going?
It's wonky.
It's kind of a mind trip, right?
Yeah. I was like, I'm fighting the urge, even though there's a camera right there. I'm fighting the urge to like turn my head because I'm just used to when I back up. And anyways, you're backing up in a car or something.
Yeah.
So you're like, oh wait, no, there's a camera right there.
This is really wanting to fight. Come on. Wow. That's cool.
Is it caught, you think?
Yeah.
Is it tied to the tray?
Not
no,
to the tray,
it can't
no.
be. Yeah.
It shouldn't be.
Yeah, make it bubble, like a different angle.
Thank you.
I need to lift up.
Why is this not staying on? Try the jaw lock like 3 times and it's not doing its thing. You want to write down that grip lock no longer works right now?
Copy.
That's fun. Or I can write it down actually.
Yeah. I'll write it down in a second. It will be noted. We offer tribute.
To Walteria.
Now it's working. That's really strange. Sticky.
Did you say we offer tribute to Walteria?
No, it's not.
Yeah. Like I said, I offer tribute. You know, Ontario was a...
Nice. That got me.
I think so. There's this position here that the grip lock's not working on, and then if it's flatter, it's working. So the cable is getting pinched. Cool. Go ahead.
That's middle.
Yeah. Nice.
Cool.
Nice.
Yeah, don't break yourself.
All right.
Let's see. Oh, that's much better. Ish. All right, moving forward, yeah?
Yep.
Bridge nav, another step.
And you see that the pan and tilt drifted again to the left, so do you a bump to the right.
Yeah, thanks.
Yep.
All right, calibration complete.
Several bamboo skeletons around would suggest that this is not necessarily the healthiest spot for corals at the moment.
Why do you think that might be?
There could be a variety of reasons. It could just be that when they started growing, there was a just slightly different current flow pattern, and that's now gone away, so they're not getting enough nutrition. Could be a herd of sea stars moved through, and there was a large predation event. Or it just could be something like a low oxygen period took them out. So it's hard to say, but there's definitely a lot of these skeletons and bases around in this area without a huge number of living colonies.
Okay.
Oh no. I think, can you pan down on the Herc, the Zeus? I think it was too high, that's why I'm like freaking out. There you go. Yeah, that's weird.
There's a big old, is that a brittle star or a crinoid?
Right here?
Can you get a zoom on that guy? Yeah.
I think it's a Brisingid star, but I can't really
Brisingid.
tell.
You can zoom in a little, up to the lasers.
Zooming in.
Nice.
Looks like a crinoid.
Bridge nav, another step.
All right,
A
zoom out.
nice little
Zooming out.
heteropathias and another plexaurid.
Full wide.
Here's another bamboo skeleton, it looks like.
Oh yeah.
Oh, nope, it's only partly up.
That's in our bumper bars. Go ahead
Yeah.
and back up. Nice. Now follow it up. See how tall this puppy is.
Oh my gosh, it
Wow.
just doesn't want to stop.
Wow.
About 4 meters,
There's
a little more.
check.
Nice.
Highway
you want to increase the speed there, Chris?
Danger zone.
0.3.
Okay. Bridge nav, can we increase speed to 0.3 knots?
Thank you.
We're gonna be cooking with gas now.
I don't know, charcoal at best.
Give us a lot of work factors.
All right.
Nice fish to the top left.
Yeah. So you're going to be at the end of your leash there. So
Yeah.
start moseying around, seeing what you want to see.
It's interesting, this area is kind of dominated by bamboo and Walteria sponges. Oh, there's a big coral actually in the back, top left.
Yep.
Mm.
I think this is a pronoid. Another calyptophora.
So for one that's this large, you can also like a little bit up. It frames it up so you don't have to fly up in the water column.
Whoa, what just happened? Oh, did you move it?
Yeah, I
Okay.
did.
All right. Okay.
But go ahead and back away from it. And then watch your butt cam. So you're on the...
Oh, yeah, I see the rocks.
Yeah. So go ahead and rotate to the right. Like a pirouette to the right.
Whoa.
Mm-hmm. There you go. I'm going to need to come up on my delta a bit because there's a rock in my sonar. So I'll have you mosey on to the left.
You got my lateral to the left?
Negative. So go ahead and stop that lateral move because you're up against the Niskins. So go ahead and move back to the right, but then pan your head to the left. There you go. So then I'll have you kind of stay a little bit upslope of me so I don't have to come down on the cliff or on the hill.
Copy.
Got a few giants on that little lip.
Yeah.
Oh yeah. Oh,
Gross.
nice. Gross.
Like one of those like long fingernail things.
Ew. When you put it that way, it is gross.
I don't suppose we could take a Niskin bottle right in here with all these pronoids and bamboos.
Yeah, what altitude do you want Michael to be at?
Anything within about 5 meters of the seafloor should be fine.
Roger. Okay, so go ahead and come to 5 meter altitude and then we'll take your Niskin. I'm gonna rack back for you. All right. Great. And then I'll drive the camera if you want to maintain a stable position.
Yeah.
did you do? We're looking at 3, huh?
We just used 3 actually. We're looking at 4.
We did? Oh, man. Are you sure about 4? Or?
Yeah, no, because we did
We're going
654.
backwards.
We're going backwards.
Yeah.
Oh.
We've done 3.
Oh, yeah. Wait, we've used 1, 2, and 3 so
Oh,
far?
this is not
We've
slurps.
used
This is Niskins.
6,
Oh, gosh.
5, and
Okay,
4.
that's correct. I was looking at the slurps. Sorry, everybody. We can use 3 now.
Roger.
It's the end of the watch.
Yeah, you know, we gotta get some food in our bellies pretty soon, too.
I need to run around.
All right, we are at 5 meters.
Roger. And fired.
Fired.
Thank you.
Yeah. Nice. Nice position holding. Awesome. You want to get centered up on the camera? I will.
Are you hobbling over there?
All right, go ahead and decrease your altitude, though. I'm gonna look up a little bit for you there. Nice. And
Yeah, it just got super dudery.
yeah. So, since the slope is kind of right downhill in front of you,
Yeah.
go ahead and rotate into the slope and then lateral down.
Actually, do I want to go this way? Yeah.
Nice. And because I'll come down a wee bit, but you won't be able to book it down slope because I won't be able to follow as quick.
Typical.
Bridge nav, another step.
Typical, typical, typical. Good. Yeah. Nice. Nice. That's good. That's looking good.
Is that a little sea urchin guy on the bottom?
My money's
I think
on
it's a
mushroom.
coral.
Oh.
Yeah, I think that's a mushroom coral.
very
Anthomastus. And lots of these big primnoids got off the crest and suddenly they all appeared.
Grab this really. Not that. Okay.
Zoom in a little.
Little zoom.
A little bit more.
A little more zoom.
Wow, I love it so much.
It's got a dense one. I don't know if it's all retracted or if it's just has a lot of arms.
think this one has a lot more arms. Asako was saying earlier that there
Zoom in a bit
are
more.
a
A little
couple
more
different
zoom.
species that have been around.
Nice.
Well, it is adorable.
All right, full wide.
Full wide.
I won't really be able to come down yet, so
Yeah.
I'll have you stay at that depth contour for a minute.
And plus, we're going to be turning over soon anyways.
Yeah, it's all on them. If you want to park it in front of a nice coral for the handover.
Yeah.
Scenic route. Thanks, guys, for the good watch.
Thank you. Nice flying.
I saw it. I saw it. I saw it. All right. Doing a switch over. And hope everyone enjoys the next watch, wherever Dan might be. And yeah.
Test 1, 2, test 1, 2. Oh, that's loud. What are we doing? Let's
All right.
see if Herc floats.
Yeah, we're taking a peek at these big corals. I guess before we go, we can zoom on them quickly.
Sure. Zoomy zoom. How about I just get a little closer? Be a good opportunity to dial my Z bias. You can zoom in there, Dan, if you can hear me.
Yes, sir.
Give this a follow-up zoom. Full. Give it all you
Full?
got.
All right.
Yeah.
How's that?
That's a nice way to start the shift.
Yeah.
Too close, Steve? You want to zoom back up?
No, I like it. It's good. It's helpful.
Can you hit the button over there?
Let's start with some checks here, Dan.
Right.
Can we drift down towards the bottom more?
Down towards the bottom, right.
It's kind of a cluster of
Can't get
something
too far
right
down
there.
or I'll stab myself.
Yeah.
Hitting bubble camera with it. A big coral. I have to back off a little bit.
This is definitely the kind of handover I like.
Yeah.
Right in front of a giant coral.
You can zoom in. You can zoom in there a bit down on the bottom. He wants to see the branching there.
There's some sort of gall or something right here.
Oh yeah.
Where is that?
Just out of the frame up.
Oh, yeah. Whoa. Bad day.
And where do those gulls come from?
Good question. It could be any number of things like parasites that are overgrown
Sorry.
by the coral eventually. This
It
doesn't
always
look
takes
like
twice.
anything living right now.
Will it eventually regrow that?
Over the branch or like over
Or
a new branch?
around it? Or
Yeah, that's probably what is happening or has happened. There's some tissue right here, the polyps that have overgrown whatever this is. So most likely it's already happened. Yep. All right. I think we're all set here.
Roger.
I see some evidence of possible grazing, but no active predators. That's one thing I'm keeping an eye out for the next little bit. General plan is continue to the southwest.
Southwest.
Towards waypoint 4.
Right.
And it's expected to be probably rocky, not very high relief, maybe some boulders here and there, but it'll be relatively gentle slopes from here through the end of the dive. But I'm hoping we maintain this biology because the currents in this area and the substrate is very favorable to having big biology.
Yeah, please. Oh, let me look around here for a minute and see what's going on. I don't know what's behind me or what's uphill or downhill. Looks like southwest is down the hill. Oh yeah.
Lynette, can you give us the layout of kind of the dive site and a refresher of where we are on the big scale of the feature?
Yeah, sure. Let's see. I don't know if our viewers can see the HiPAC screen. If it's up, I'll zoom out so we can get kind of an overview of where we're at.
We can, Dan can put it up on one of the feeds.
I think I got it. Out there.
Oh, that looks like the sonar one.
Oh,
I think.
nope, sorry.
Okay, so
So if
There
you're
we
watching
go.
on channel 3, or if you're on YouTube, go check the channel 3 feed to watch this dazzling display.
Look at that crazy, crazy bathymetry.
So we started on a ridge up to the north, and we're following that ridge kind of southward until we get to the summit. So you can see where my cursor is here. We're near waypoint
Roger.
4.
Can you spin around and look southwest?
And we'll be continuing up onto waypoint 4, and then upward with a pretty gentle slope, I think, all the way to the summit.
Firecracker coral.
Is this a conical seamount or a
I'm going to go the other way around just to get rid of the wrap in the 6-8.
Yeah, go clockwise.
Clockwise? Okay.
Or whatever, take that.
Yeah,
half
it looked
wrap
like
out
a
of
sharp
the-
peaked seamount
Yeah.
when we planned the dive. But the top is a little bit more flat, but not like yyyy flat.
Jeez.
There's a bit of a platform on top, but it's not clear if it's carbonate or not.
So what classifies a guyot versus the seamount?
Heading 240, looking southwest.
Well,
Ooh,
let's
look,
ask
right
our
down the
geologist.
spiral.
Brandon?
Nice.
So
I don't know
guillemot,
if you can
a
get that
guyot
shot.
is just, it has to be flat topped. There's no like distance range as far as I know. You know, sort of a parameter for it, but it needs to be rather large, like
you pull up the
several
DSC?
kilometers of flat top. And we don't see that on this seamount.
So if we have a little bit of flat top on the seamount, it's not considered a guyot?
No.
Okay.
I'm going to try and get look down the spiral.
It could be
with
the resolution of the
DSC.
bathymetry too.
Yeah, that would be my guess.
Yep.
Is the I think these are 200 meter or no 100 meter
So
contours.
looking down the spiral there, and I got to come
So-
up a
Just
little.
about.
Yeah,
Come up a little bit.
the grids are usually plotted at 50 meters, something like that.
Come up this a little.
Love it. So the DSC camera is
Right there.
mounted on the porch.
Sweet.
It is a still
Sorry.
camera
Nice
that
shot.
is being
I don't know why that tickles me.
controlled by the back row. We're experimenting. We have on many dives just left this on the intervalometer, which is one of my favorite words we get to use here. Just taking photos every 30 seconds, but now we're
Cool word, horrible photos.
many photos, many
work.
many photos.
Much work for you, Megan, eh?
Yeah.
More better this way.
I feel like it's kind of like replay though, and in the other manner, I get to like
Yeah.
see every landscape.
You can say this way, you can say we didn't use the intervalum meter. We actually got this. We worked for it.
Do some people say interval meter instead of intervalometer? Am I saying it weird?
Jonathan says it intervalometer, but he's
Oh.
a total geek. He's like a photo geek. Well, he's actually a video geek, you know.
With love.
He doesn't care much about photos, so.
So we've been seeing kind of similar species over the past, let's say, a couple hours or so, since we got up on top of this local high within the ridge, small cone, right around waypoint 3. But you know, I think what we're seeing today is different in many ways, owing to the depth differences that we are between yesterday and today.
Okay,
We're
that.
a little bit shallower.
Okay,
We're somewhere around
What are we doing? Let's do
1650
point
meters,
two,
which
'cause
is
I'm going
right
really
around where
slow
we ended the
backwards.
dive yesterday, but the seamount still continues to
Point
go
two,
up
yeah.
at this site, which would give it access to species
I
that might
can't
have
hear you at
shallower
all.
depth distributions. But we will continue upslope and see if there's any change in
Which is
community
strange, I'm
composition.
listening.
Could be.
Oh, you're turning
So what do
way
you think is
down.
going to be
What
the most
heck?
common life form that we see up here?
Yes,
Or
ma'am.
down here, rather?
Corals and to a lesser degree sponges on top. Yeah.
Sure.
So
But-
Steve, I know it's hard to do this mid-dive, but have we been seeing a different species already at this depth than we were seeing on some of the other seamounts?
At this depth, yes, because we haven't really visited this depth on many
Okay.
other sites. So we're seeing a lot of large primnoids, many different species, a few different genera that we've observed at other sites. Things tend to look a little bit bigger here. I don't know if it's because of just the particulate matter in the water column. It appears to be a little bit more turbid, which could be an indicator of good food supply for corals this depth. There's also fairly stable substrate. It's not rubble or talus material. They're probably going to be able to stay stuck here for quite a bit. In fact, you can see those small halos on the rock surface where corals previously lived.
So odd shape, but I don't. You see predators on that guy?
No.
Okay, moving on.
And I think that's a bamboo coral too.
You
We're,
want-
we're kind of, we'll take what we can get, but we're preferentially interested in primnoids like the one we saw when we first got on.
Okay.
But we'll keep an eye out. I think sea stars will probably be the easiest predator to look for.
If you say primnoid 20 more times, I might get it. I might
Primnoid,
make the
primnoid,
association.
primnoid.
No, you have to say it while I'm looking
Oh.
at it.
Okay.
You know, I can burn it into my little reptile brain.